we will start the revision this week. Okay, so I just shared today. Most of I do not know also. So we will have the revision this week. I will send the file to you, the schedule. But today we have topics 1 and 2. Okay. Tomorrow, Dr. Chung, for tomorrow is 4th September. 4th of September. You will have a session with Dr. Chung in the afternoon. Okay. On Friday is this topic 3. Dr. Narabasana. Next week, Tuesday, is topic 4. This will be online by WebEx. And this is topic 6, the structural mathematics 2. Okay, so I will send this to you after this.
Yeah.
After the class.
The training 2 weeks.
Yeah.
Training 2 weeks.
Yeah, training 2 weeks. So you will just be like this.
Yeah.
Okay, I will help you.
Totally 2 weeks.
Yeah.
And exam has not been decided by university, but they told us it's these days, 17 to 19. So it's the 3rd week.
Mhm.
Yeah, after.
How much need after C result?
Yeah.
How much need the time after C result?
Very fast because you need to register.
Oh no, doctor not need register.
No, not register, but I mean,
Yeah, I know.
for
A new semester.
the new semester for the
next
third semester.
Yeah.
So you will get the results very fast.
Please, you need help.
Yeah.
Together.
Okay.
So, not decided the
Date.
Yeah.
Yeah, not yet because university will decide.
Oh.
We cannot decide. Yeah. But they tell us in these 3 days, so one day they will decide.
Okay.
Yeah. After they decide, we will tell you.
Yeah.
Okay, so you have this.
Yeah.
Okay. Did you read the question first? Please, 10 more questions. Okay. So you have DNA sequence,
Yeah.
and DNA sequence contains a protein coding gene from bacteria.
Yeah.
How do we find this protein coding gene? So we use the ORF finder. Okay, so if we use the ORF finder to Google, okay,
Okay.
yeah,
Okay.
ORF finder,
Mm.
ORF finder, and then you have this NCBI result, ORF finder. Okay. Finder. So the first question is, what online program do you use to analyze the sequence? Do you see the question?
What's on? To use some.
Yeah.
Well, Mark.
Yeah.
Like that.
So we need to know the name of the program and also
Yeah.
what is the function of the program. So ORF Finder searches for open reading frames. Do you know what is an open reading frame? An open reading frame is a gene. Right?
Yeah.
So in a DNA sequence, there is a gene. So for example, here, this unknown sequence is 2,280. This one is 2,880, but somewhere inside this sequence, there is an open reading frame or gene.
Yeah.
So we use this software to help us to find the open reading frame. Okay, so the open reading frame, there are many predictions. Okay, you can see here. 12 predictions, right? 12 predictions. 12 predictions. So they predict start codon ATG. Do you know what is a start codon? Do you understand about start codon? So start codon is the
3.
sequence that is recognized by bacteria or human or different types of cells to indicate it is the start of a gene or an open reading frame, we call it open reading frame, ORF. So the gene is the start of it, it's ATG.
Okay.
So the start calling is in front of the.
Yeah, in front, yeah, of course. So because in the organism, in the bacteria, in the any cell, like for example, we have a cell,
Mm-hmm.
and inside here is the nucleus,
Mm-hmm.
and then a lot of DNA, and DNA
Double
is
sequence.
very long, right?
Yeah.
There is no start,
Double
there is no
sequence.
stop. So how does the cell know where is the start? Right? So to know this is the start, so usually it is APG start,
Mm-hmm.
because there is no signal. So this is the signal that is common APG. Then where does it stop? It would be A. So there are a few sequences to start and
So
stop.
start and stop.
Yeah,
Sequence.
yeah, start and stop.
In DNA strand.
Yeah,
In DNA strand. Any part in DNA strand.
yeah,
Start ITG. Stop
yes,
TG.
yeah. So you have APG here,
Yeah.
ATG. So the software finds where is the ATG, right?
Mm-hmm.
G. So if we look here, there are many ATG. See, right? There are many ATG,
Navigation.
right? So there are many ATG in this sequence. Of course, the software will find ATG with um,
Mm-hmm.
TAA. It's the stop sequence.
Mm-hmm.
TAA. Okay.
Tail PGE.
PGA.
PGA.
Yeah. So it can be TAA or TGA as well. So it
Oh.
depends on the sequence. So there are few recognition sequence.
Depending the sequence type.
Yeah, it can be recognized by bacteria or human cells or animal cells.
Mm-hmm.
Yeah, so different in bacteria there are several stop codons and several start codons. Okay?
Mm-hmm.
So start codons here, for example, is ATG. So it's written as U because in the mRNA there is no T. mRNA consists of uracil. So you know that DNA has 4 bases, right? DNA.
And they have 5.
4 bases. A, G, C, T.
Yeah.
Adenine, guanine.
Adenine, guanine, cytosine, thymine.
Yeah, cytosine and thymine. So A, G, C, T.
Yeah.
Right? But in mRNA,
Thymine, adenine, guanine, cytosine.
there is no thymine.
Superstructure.
There's no thymine.
No thymine.
Becomes uracil.
Uracil.
Okay? So that's why they write it as AUG. Okay, but usually we look at the DNA sequence. Okay, we look at this. But anyway, here you have AUG, and also you have other start.
Start.
Yeah. So stop codons can be
3.
yeah TAG, TAA, or TGA.
Okay.
Okay, so these are
Uracil, adenine, thymine.
yeah different types of stop codon.
For DNA is.
Yeah, for RNA. So because when we make a protein during protein synthesis, what is being processed is mRNA. However, mRNA comes from DNA, right?
Okay.
So when we sequence,
This messenger.
yeah, messenger RNA, but the information is stored in DNA,
Mm-hmm.
and we sequence in DNA.
DNA, RNA, same sequence?
Yes, the same sequence. Only the difference is U.
I know.
Okay, but in the database, So as humans, we keep DNA information.
Specific.
Yeah, because we know that in RNA, you only change from T to U. Okay, so in all the database in the world, we keep DNA information.
Mm-hmm.
All right.
Correct.
So that is why we see T in the sequence. This is a DNA sequence.
Okay.
So based on the software prediction, okay, for example, you look at here, there are many ATGs, right? However, ATG
Many
must
start for
have a stop codon, and they must be in-frame.
you can
free.
choose a small part for DNA.
Yeah, so it must be codon, ATG, and then 3 nucleotides, then 3, until we reach the stop codon.
Mm-hmm.
So that is why open reading frame find the primes to predict, okay, what is the available sequence inside the unknown fragment. Okay, so
Mm-hmm.
you have this, and so on. So for example, if we take this, then the first it starts from 569 to 2056.
This during ask find the ORF.
Yeah, ORF. So you will predict this first.
So you see the population, start population,
Yeah,
the number.
yeah. The number is start from 569. So if we take this, okay, see 75.
Check number 4. Please.
Okay. So here is okay, so one line is 60, 60. Right? It's number 1 to 16.
60.
Okay. So if I say 5, 0. So this is 60. This one is 150.
Oh, it's big.
Yeah.
Edit 60 all the state.
Yeah, they are the same. 60 to 40.
Mm-hmm.
60.
60 for
40.
What wondering me?
this you can find the number minus in a question, final question, take the 2 number. You can, how can you find ORF? First one minus first 2, you can find this number, this ORF. Okay, before the show.
Okay, so this is 600. So this is 569.
Uh-huh.
569. So this is the start position. Number 569 is here. Okay, so it says here 569. Okay,
This is start.
yeah, that is the start. So let's put the one.
Start.
569. So this one is 569.
Okay.
A is 569. Do you understand? Okay. 569. And the last one,
Start. 200. 2000.
yeah, the last one is 2000.
What?
Yeah.
Two thousand.
2049.
49, yeah.
2049.
2056.
2056.
Yeah.
2056.
56.
Okay. So, because we want to first calculate the length.
You need to find the
Yeah.
ORF.
RF. So the RF says that 2056 and 569. So this is the length.
Yeah.
1488. This is how
Between
long?
them.
Yeah. How long is the open reading frame? Okay. So take 2056
Minus
minus
569.
8 because 568 because 569 is the first
Okay.
first one. Okay. This is 569,
Mm-hmm.
but you have to remove this
Okay.
568.
68.
Okay.
Yeah,
But in
because
question, take the in
Yeah.
question 568 or 569. In
Yeah,
question, for
568.
example.
yeah, so it's this is the length
Okay.
140.
Okay.
So to check this whether you are right or wrong, you have to divide by 3. Okay, divide by 3 because one codon is 3.
Mm-hmm.
What is this?
So
Let's find the color.
let's do this again. So in the question, okay, first position is 569
Mm-hmm.
and last position is 2056. Okay.
Mhm.
First position 569.
Already take first position and last position in a question.
Yeah, 2056.
In question find is last and
Yeah,
but need how make the ROF the 2000.56 minus 568.
yeah. Okay, so this is the length. So because just now we are discussing the exam question. Okay, so this is the final exam question that you answered. Okay. So this is the
This
question.
step 1, step 2.
State the nucleotide length. Length means how long is the okay. So 5035.
This start 505 start.
Yeah, 505. 505 start.
Yeah.
And 5035 is the last. So we take 5035
Minus
4504.
this ROF.
These are RF. Yeah.
Mhm.
Okay, so RF.
What is OF?
RF already
Given.
given.
Yeah. So if you divide by 3,
Mhm.
you get the full number is correct. So 5035
Uh-huh.
minus 4505 get this, but if you divide by 3, you get this decimal number.
This last question.
Yeah, so if you do 5035 minus 40, so in minus 4505, you will get this. So if I take
This last or how?
5035 minus 4505, right? 5035 minus 4.
Why isn't the 4504?
Yeah, no, I just want to show you.
Oh,
Okay, so you get this answer.
yeah.
If you put 530 here,
Too much.
it's the wrong answer. It's the wrong answer because
Yeah.
you cannot divide by 3. Okay, you cannot divide by 3 because open reading frame is always can be divided by 3 because it's a codon. It's a codon, right? And one codon codes,
Oh, I know.
okay?
Yeah.
One codon codes for one amino acid. Okay, so that's why we have to go to the next question. What is the gene encoded in the ORF?
Bacteria.
And what is bacterium stood for? Okay, so to do this, we can do a blast.
We start.
Okay, we can do a blast. Okay, so it says that it belongs to E. coli or Enterobacteriaceae. So
You
E.
name
coli.
the gene in
Yeah.
question.
So in the question, it
You
says,
name the gene.
yeah, what is the gene
Uh-huh.
encoded? What
Already
bacterium?
E. coli.
Yeah, so the gene name is TOL-C, family outer membrane protein.
Where find this in the question? Not find.
Yeah, I said outer membrane channel protein.
So you can go in the question.
Okay, the question is what is the gene?
Up. No. What is the gene encoded?
Yeah, so now we are looking at this question. What the gene encoded in the ORF?
How can now
Yeah.
the gene is bacteria or human or
Yeah.
something? How can find?
So first, this is the prediction, right?
Yeah.
For the frame, and then we need to blast to check.
I don't have laptop during the exam.
Yeah, so we will not ask you this kind of questions because we
Mhm.
cannot check. So we will give some information. Yeah, or maybe we can ask you, what do you do to check? So what is so if I ask you, how can you check for?
Copy the sequence.
Yeah,
Go the NCBI check.
yeah,
You can the NCBI check during the result can find the gene,
yeah,
how the organism.
yeah. So you must say that we can use blast, for
Yeah.
example. So of course we cannot ask you to check during the exam
Mhm.
because
You can write the check, open the e-learning, open the NCBI,
yeah,
check the ORF,
Yeah.
NCBI, copy the sequence in
Yeah.
this. You
Yeah.
can write this step
yeah,
for this question.
yes.
Okay.
Yeah. So we want to know that you understand how to
Mhm.
find the information. Okay. So you
You
can say.
understand?
So you can say
I
we
think.
can use blast to
Mhm.
find the name of the gene, for example.
So,
Okay.
what is the difference with smart blast and blast?
Okay. So smart blast is a quick blast
Specific.
Oh,
because the database is very big, billions of sequences. So if you do blast, they will do a full search. But
Full
sometimes,
search.
yeah, full search means that they will search all the database. But smart blast, they will only find limited comparison. Do you know how the blast works? So blast is when you have a DNA and you want to match the database, right? Okay, so you have open reading frame here. You can ask it.
What difference between plus? Small plus.
Okay.
If the bus number two, specific data. You specific data. So
So
different
Smart
general.
blast is a faster blast because you only search
A
for
specific small plus.
limited information. For example, using protein search, blast P. P is for protein. Okay. So coming back to our question, so what is the gene encoded?
Equal
And you can write here. The gene encoded, for example, outer membrane protein. So this is the name of the gene.
12. Okay, energy.
Outer membrane protein channel protein. Okay. What bacterium is it from?
E. coli.
Yeah. So the bacterium, it's because
Two bacteria.
the
Escherichia.
summary of the results shows that unknown, it is the unknown, right? That we are searching. It is closely related to E. coli. This is the unknown.
You
So,
can write E. coli or
yeah. You can
only E.
write,
coli.
yeah, so you can write as Escherichia coli.
Escherichia coli.
Okay. And we must write in italics for species name. Okay.
Okay.
Species name must be italic.
Okay. Question 3.
Okay.
What other
What
information?
other information can you obtain from a DNA sequence of the fragment? Are there any regulatory sequences upstream of the open reading frame?
Sorry, I
Hey.
don't.
Yeah.
ATG? Open the reading frame for.
No. Okay. Later I will tell you.
Last question, I don't very clearly.
This question.
So,
Yeah. Okay. So what is the gene encoded in the ORF means that, okay, the open reading frame finder has predicted there are a few ORFs. They have predicted a few ORFs. So they have predicted ORF1 here, ORF3, ORF10, and so on, according to the size of the ORF. So they have predicted a few. So we start with the longest one. Okay.
yeah,
So we can also check this. So this is
The dictum.
yeah. This is the next ORF that it predicts, ORF10. So we run a smart blast. ORF10. So there is no results, right? One blast found no matches because it is not a true ORF. Okay, that means that it does not exist in the organism. It is just a prediction, but it doesn't exist. So it's not a true ORF. So from there, we know that the first one is 569 to 2056, length of
This is
1.
not for ROF.
Yeah. So this is the true ORF, right? True ORF because it is, when you blast, you get a result because it can match something in the database. So you have unknown and the database is here, so it can match. You can find the database. Just like maybe criminal, you can search a face. If a robber, do you know what's a robber? A thief, you know a thief? No?
robots.
Yeah, thief. Or killer, do you know what's a killer?
Yeah.
So a killer, right?
Yeah.
A killer, so he's a criminal. the police can search
Ah,
the face of the killer
yeah.
if it exists in the database, right?
Mhm.
So if you have the killer's face, then you can check the database.
Uh-huh.
Okay, this person exists.
Only database different in other database because only database have the killer face. Have
Yeah.
something is
Yeah.
not.
So the same here.
Yeah,
In this case,
yeah.
open reading frame 2, which
When-
is for F10. In the smart glass, it does not exist, so there is no information about this.
Okay.
Right? There is no information, so this is not a true open reading frame. Next, we have other ones, and if you check all, okay, you can check glass, check
Plus.
glass, so you will find no results because they are not the real open reading frame. It is not a true open reading frame. Okay?
Okay.
So we know the first one is that this exists because there is information in the database. And this protein is called Tolsi family outer membrane protein.
This for bacteria.
Yeah.
It's
It's
coli.
from E. coli. Our unknown open reading frame is very similar to E. coli. As you can see here, 100% identity. So it's 100%. Right? 100%
Yeah.
means they match. Okay?
Yeah.
If you look at this species, Pseudomonas.
Three types.
Yeah, Pseudomonas is here. It does not match. You can see. Okay. So here is our unknown protein, unknown open reading frame, and this is the
Database.
Pseudomonas in the database. Right. So we have some of it is. Let's see which one is the same. A is the same. A is the same, right?
Mm-hmm.
L is the same. B, Y. But here it's not the same Q, but here it's Y. Okay, so they are not identical, and they are only. So they are only 83. No, they are only 28% identical. 28% the same sequence.
Similar something.
Yeah, so the identity is very low. Here it's very low. But 100% it's E. coli.
So we must choose the 100% one.
Yeah. So we choose the 100% one. Okay.
So this is the first question.
And
This is what is the gene code in the ORF, and
why is bacteria?
yeah.
So it's
Uh,
called the outer membrane protein.
so the name of bacterium, where?
Yeah. So name of bacterium is
Greek?
Enterobacteriaceae. Yeah, you can see here. Enterobacteriaceae. Multi-species. So it means that it's found in many bacteria.
And
And you can also see here E. coli because unknown is very near to E. coli, as you can see in the alignment.
I don't know.
So unknown
Yeah, I know.
is from our open reading frame finder.
Yeah.
Known result.
Yeah, it's the unknown result. So they are checking the unknown.
Yeah.
The unknown is inside this group.
Yeah.
It's
Yeah.
similar.
Yeah, so it belongs to the E. coli.
Bacteria.
Belongs to the E. coli species of bacteria. You can see it's nearest to here, and it is 100% coli.
Oh.
Okay, and it is very far from here. Share one about 80%. So Shevanella, not 80%, but 42% similar only. Okay, so it is not the killer. It's, maybe the killer's cousin. The face is not the same,
Mhm.
right? It's relative.
Same something.
Same something, yeah, because they are very close. And maybe not brothers, but they are
Mhm.
family member. Yeah,
Yeah.
so 40 something. This one is 28%, so it's more distant family member. Okay, so we know that it's E. coli from here, because it's very near this group.
I just want to find the name, the ECIL.
Yeah, Escherichia coli.
Where?
This one,
Ah,
Escherichia coli. Yeah.
could you那个返回啊为什么 This is what page?
Sorry?
What page?
What page? Okay, so we do the smart blast.
What?
We need for question.
Yeah. Let's wait.
This way,
Turn.
Oh,
Oh.
Three questions.
ESCHERI.
The bacteria. Ask me all the bacteria.
Yeah, this is the name of the bacteria.
You can
ESCHERI.
write the Escherichia coli or the.
Yeah, so the bacterium is Escherichia coli.
Mhm.
Yeah, this is the bacteria name.
So we also can use the Eton. This one name. You know, this one name. We also.
Enterobacteriaceae.
Yeah.
Same.
Yeah.
Oh yeah.
Enterobacteriaceae
So
is
Tom.
the
family.
the
Yeah.
family.
Yeah.
Oh.
It's the big family.
The E. coli have too much type.
Yeah, so much like so it's called Enterobacteria.
Me, I work with a 3 type E. coli.
So
DL21, DE3, top 10.
yeah, the E. coli is a common bacteria, so it's inside our body. It's a common.
Some E.
bacteria.
coli normal flora.
Yeah, normal flora. But it's a common bacteria, E. coli. Okay. Yeah. Also in our lab, like you
Yeah.
mentioned. Yeah. Okay. So the next question is: are there any regulatory sequences upstream of the RNA? Okay, so do you know what is a regulatory sequence? Okay.
It's 14. Promoter.
Yeah. Do you know what is a gene
Yeah.
promoter?
Yeah, I know. Me, I know. Some bacteria have 3 promoter. This bacteria, not DE3. The bacteria have 7 promoter RNA.
Yeah.
Bacteria DE3.
DE3, yeah. So this is a very common promoter in the cell. For example, it's called lactoferrin.
Mm-hmm.
Hi, you want to join us? So how the lactoferrin works is there is a DNA recognition sequence, DNA sequence, in front of the open reading frame. Okay, so there is a promoter in front of the open reading frame. So you understand what is open reading frame now, right?
Yeah, yeah.
It's a gene, but how do you control the gene to make sure that it is only expressed when it is needed?
But
When the cell needs the gene, then
Yeah.
only it is produced. Okay, when it is being synthesized to make mRNA and to make
Yeah.
protein. So this is where the regulatory sequences come. Okay, so you have the regulatory sequences. So
DNA, RNA,
Yeah.
promoter.
Yeah, so there is a promoter in front of the open wheeling frame. Okay.
It's used for shake promoter G.
Yeah, so check the promoter G. So promoters are located
Copy all the sequence.
in front of the sequence. So we call this as upstream.
Mhm.
Upstream
Yeah.
means in front
Yeah,
of
yeah,
something. Upstream. Okay, so I put this here.
Hot. This captain.
I know this because my plasmid have the connect to the
Try to rather. Take all of them.
I think the internet not good. You are okay?
What we do not.